Hi everyone, welcome to my channel Pure Biology, and I am Tapushi Karmakar. And today's topic is the variation in the chromosome number. Okay, so what is the variation in chromosome number? That means number of the chromosome may be changed, either decrease or increase due to the chromosomal mutation. And before going to topic, I just want to say that this chromosomal mutation and gene mutation, actually I explained in my previous video. You just check out what is the chromosomal mutation properly. Okay, and I described properly in there, but here I don't explain about the chromosomal mutation. I just explain here what happened if the chromosomal mutation occur. Okay, how the changes occur in the chromosomes, number of chromosomes. Okay, so here first we discuss about the variation in the chromosome number. You can see when an organism or the cell that has the one complete set of chromosomes, or an exact multiple of the complete sets of that organism or cell is said to be euploid. Okay, that means if one complete set of chromosome is present or an exact multiple of one complete set. Okay, if one complete set or multiple of one complete set is present, then in that situation it is called the euploid. Okay, so eukaryotic organism, you all know these are diploid. Okay, eukaryotic organisms generally diploid, but eukaryotic organisms which are the haploid, they are generally euploids. Examples of euploids. Okay, so chromosome mutations that create the variation in the number of the chromosomes and that resulting the formation of the euploids. If the chromosomal mutation is not occurred, that situation no number of changes occur in the chromosomes. But if the chromosomal mutation is occurring, that means the euploid chance of euploid formation is very there is possibility, maximum possibility for the formation of the euploids. Clear? So here are the three terms that are explained, that aneuploidy, what is autopolyploidy, and the allopolyploidy. What is called the aneuploidy? Aneuploidy means one or few chromosome loss or gain here. Okay, one or few, number of one or few chromosome loss or gain is seen here. But here involvement of change in number of sets is not seen. It is very important. The number of change in the sets is not seen. Okay, the set, the number of chromosome number may be increased or decreased, but set, number of sets is remain same. Okay, and this is generally possible due to the non-disjunction process. Okay, that non-disjunction process occurs when during the meiosis or mitosis situation. Okay, and the two term is very important. One is the monoploid, another is the polyploid. What is called the monoploid? Monoploid means when the chromosome is one set. That means one set of chromosome when present instead of two. Generally two sets of chromosome must be present. So here you can see the one set of chromosome is present. That means it is called the monoploid. And which one is the polyploid? When more than two sets, more than normal two sets of chromosomes present, then it is called the polyploid. Clear? So this is monoploid and this is polyploid. Next one is the autopolyploidy and the allopolyploidy. Autopolyploidy means here the all sets of chromosome that originate. Okay, suppose these are the sets of chromosome. Okay, these are the sets of chromosome. This is one set and this is another set. Okay, so you cannot see. I think you cannot see. Okay, suppose this is one set and this is one set. So two sets of chromosomes. If the two sets of chromosomes that originate in the same species, okay, this is originate in the same species, then it is called the autopolyploidy. And when it is called the allopolyploidy, when the sets of chromosome involved come from the different though the related species. When the two species involved but related species, when the two species involved, in that situation the sets of chromosome come from the two species. Okay, so this is called the allopolyploidy. Clear? And here in case of allopolyploidy, the two different species interbreed. Two different species generally interbreed. Okay, and the sets of chromosome come from the two species. Clear? And in case of autopolyploidy, you can see the diploid and the haploid. Okay, diploid and haploid gametes you can see. And when the also not haploid, diploid and triploid gamete you can see. And triploid, how is possible? When the diploid gamete that fuses with the haploid gamete, then the triploid gamete, chances of the formation of the triploid gamete is possible. Okay, so these are the terms: aneuploidy, autoploidy, and the polyploidy. And next I discuss about it briefly. Okay. So this is the types of the aneuploidy. You can see these are the types and these are the formula. Formula means what happened to in case of number of chromosomes. Okay. So in case of nalisomy, these are first types you learn about it. The numbers of chromosome with 2n minus 2. That means it's called the nalisomy. Okay. When the nalisomy, what is the meaning of the nalisomy? In that situation, you can see the loss of one homologous chromosome pair. That means here the two homologous chromosome totally deleted. Okay, homologous chromosomes that totally deleted. And in that situation, that sets what is the type of set we can see, that is a 2n minus 2, and this is the formula, or how to represent the nalisomy. By this formula we can represent the nalisomy. Clear? Second one is the monosomy. In case of monosomy, here the only single chromosome that is lost totally. Okay, so in that situation the formula is the 2n minus 1. That means that by representing this formula we can say that the monosomic cell, it is the monosomic cell. Clear? Next one is the trisomy. In case of trisomy, the involvement, that means addition of single extra chromosome. Clear? Single extra chromosome, this is called a trisomy. And in that situation, trisomy, the formula is 2n plus 1. Okay? And in case of, that is by representing this formula, or we can say that the cell is trisomic cell. Clear? Okay, next one is the tetrasomy. In case of tetrasomy, the involvement of the extra chromosome pair, that means two chromosomes added there, and the formula becomes 2n plus 2. In case of double monosomy, in that situation, loss of one chromosome first and then again loss of one, not loss of chromosome pair. Okay, if the loss of chromosome pair, that means you can say the nalisomy, but here it's not possible. What you see that first of all one chromosome that is missing, and after that one chromosome is missing. Not simultaneously the missing of two chromosomes occur. So this is called the double chromosomic double monosomy. Okay, and the cell is called the double monosomic cell. And what is the formula? Formula is the 2n minus 1 minus 1. Okay, and the last one, this is called the double trisomy. It is the name of the cell name. Okay, double monosomic cell, and the term is actually double monosomy. Okay, double monosomy. This is called the double monosomy. And the last one, this is called the double trisomy. What happened there? Extra one chromosome first, again one extra chromosome added. Not addition of the two chromosomes simultaneously, like involvement of the single this one. Not like this one. The pair is not added simultaneously. You follow this one. What is the trisomy and what is the double trisomy? In case of trisomy, the one pair of chromosome, extra chromosome, that added simultaneously. But in case of double trisomy, not simultaneously the chromosomes added. First addition of one chromosome we can see, and then after the addition of one chromosome. Okay. So this is called the double trisomy. And the formula is 2n plus 1 plus 1. This is double trisomy. So these are the types of the aneuploidy, or this type of aneuploidy or the chromosome number variation you can see. Clear? So next one we learn about the non-disjunction. What is the non-disjunction? Non-disjunction is actually happen in case of when the meiosis 1. You know all the meiosis first division, meiosis second division. So if the meiosis in the situation meiosis 1, the non-disjunction is occurring, and then what happened? 2 with the duplicated gamete, duplicated obviously chromosome, 2 with duplicated chromosome, and 2 with this particular corresponding chromosome missing gamete. So the gametes are formed, okay, but 2 with the duplicated chromosome and the number of gametes is 2, and the 2 with that missing chromosome, that corresponding missing chromosome. These gametes are formed. In case of non-disjunction, when occurs during the meiosis 2, what happened? Here the 2 normal gametes is formed and 2 abnormal gametes is formed. Clear? 2 normal gametes is formed and the 2 abnormal gametes is formed. And here one example is that a single gamete with 2 daughter chromosome and other with 2 missing. Clear? This is the example that if this non-disjunction is occurring in meiosis 2, you can see one gamete with 2 daughter chromosomes, okay, and another gamete that is formed by this non-disjunction process is occurring in the meiosis 2. Here, the other gamete that the daughter chromosome missing, 2 daughter chromosome missing, one with 2 daughter chromosome and another 2 with missing. Clear? So these are the situation that create when the depending on the non-disjunction process obviously. If the non-disjunction process occur in the meiosis 2, this situation arise, and if the non-disjunction situation that occurs in during the meiosis 2, then this type of situation or condition may occur. Clear? And here to diagram you follow that it is the auto polyploidy and this is the allo polyploidy. Okay, so in case of auto polyploidy, you can see the transfer of chromosome is possible only one species. Only one species. Clear? But in case of allo polyploidy, you can see the transfer of the chromosome that is possible 2 species, but obviously related. 2 species. Transfer of this from here one of these, and from here one of these is transferred, and here also one of these and one of these transferred. Okay, this doubling and form this type of number of chromosomes gamete. Clear? So these are the variation in chromosome numbers. Okay, and in my next video I discuss about the disease that is created due to this the variation in the number. That means the normal chromosomes obviously 2A. Okay, but if this situation arises, then obviously that is not normal. Obviously this is abnormal, and in this abnormal situation, what disease we can see, okay, genetic disease we can see, obviously I discuss in my next video. Okay, thank you.